Bovine ephemeral fever, BEF, also known as 3-day sickness, is an arthropod vector-borne disease of cattle and is caused by bovine ephemeral fever virus, BEFV, a member of the genus Ephemerovirus in the family Rhabdoviridae. BEFV forms a bullet or cone-shaped virions that consist of a negative, single-stranded RNA genome with a lipid envelope and 5 structural proteins. The envelope glycoprotein G contains type-specific and neutralizing antigenic sites. There has been recent evidence which demonstrated that BEFV induces apoptosis in several cell lines. It was however shown that apoptosis could be blocked by the caspase inhibitor Z-VAD-fmk, indicating that BEFV induces caspase-dependent apoptosis in cultured cells. Location: the virus has been found in tropical and subtropical regions of Asia, Africa and through eastern Australia. Transmission: the virus is transmitted by an insect vector. The particular species linked to the virus are the biting midges Culicoides oxystoma and C. niponensis. Disease characteristics: the characteristics of the disease are the sudden onset of fever, stiffness, lameness and nasal and ocular discharges. BEF often causes hypocalcemia, which in turn generates clinical signs such as depression, cessation of rumination, muscle tremors and constipation. Although the pathogenesis of the disease is complex, it seems clear that the host inflammatory responses, mediated by the release of cytokines, are involved in the expression of the disease. Diagnosis: the virus can be isolated from blood and can be identified by immunofluorescence and immunostaining. Rising antibodies in 2 samples taken 2 to 4 weeks apart also indicate infection. Treatment: calcium borogluconate and NSAIDs often help to alleviate clinical signs, but rest is absolutely imperative. For a minimum of 1 week, a death can still occur. Control: a vaccine is available in Japan but is very expensive, which limits its use. Movement regulations are in place in several countries. UltraVac-BEF, from Zoetis, is a vaccine to prevent bovine ephemeral fever. The product is registered in Australia, Israel and Egypt. References: Bovine ephemeral fever, Wikipedia. Net. Retrieved 23 February 2012. Shi A. Y. C. Chen. S. H. Chu. C. C. Ting. L. J. Itakura. C. Wang. F. I. 2005. Bovine ephemeral fever in Taiwan. 2001-2002. Quote. PDF. Journal of Veterinary Medical Science. 67. 4. 411-416. DOI. 10.1292, JVMS. 67.411. PMID 15,876,792. Chang. C. J. Shi. W. L. U. F. L. Liao. M. H. Liu. H. J. 2004. Apoptosis induced by bovine ephemeral fever virus. Journal of Virological Methods. 122. 2. 165-170. DOI. 10.1016, J. J. Viralmit. 2004.08.016. PMID 15,542,140. Shi A. Y. C. Wang. S. Y. Li. Y. F. Chen. S. H. Mac. P. O. Chu. C. Y. 2006. DNA sequence analysis of glycoprotein G gene of bovine ephemeral fever virus and development of a double oil emulsion vaccine against bovine ephemeral fever. PDF. Journal of Veterinary Medical Science. 68. 6. 543 to 548. DOI 10.1292 JVMS 68.543. PMID 16,820,709.